Okay, so welcome again to our Environmental Statistics Week. And today we are very happy to have our own co-leader of the call, Dr. Sung-Hyun Park, to talk about his research on environmental cadmium and mortalities from influenza and pneumonia. Sung-Hyun Kim is Associate Professor of Environmental Health Sciences and Epidemiology at the University of Michigan School of Public Health. He is also the co-director of the Occupational Epidemiology Program in the Center of Occupational Health and Safety Engineering. So his research focuses on health effects of various environmental pollutant exposures, such as air pollution, heavy metals, non-persistent chemicals, and also persistent organic pollutant. So he recently developed a statistical approach to integrate the multiple pollutant and pollutant mixture. His lab also has a specific interest in gene environmental and nutrition environmental interactions that is very helpful to identify the vulnerable subpopulations to the environmental exposure and also to provide intervention strategies. He's working with several ongoing cohort studies, such as study of women's health across the nation, a normative aging study, multi-ethnic study, and Amish family diabetes study, and also the National Health and Nutrition Examination Surveys. So without further ado, Professor Park, the floor is yours.
Thank you, Lou. Hi, everyone. So I'm very happy to introduce my recent study about this environmental cadmium exposure and mortality from influenza and pneumonia in U.S. adults. So first, I want to acknowledge so this is a collaborative work. Dr. Howard Hu, who used to be the chair of the EHS department, currently the chair of the Department of Preventive Medicine at USC Keck Medicine. And another, the Dr. Mati Siren. So I'll introduce who Dr. Siren is later. And also, M-LEEaD pilot project mechanism, the expedite review, and then allow this project to be done. So I appreciate the funding from M-LEEaD Center. And so this paper is currently under review. So this is right now I am having the second round of review at Environmental Health Perspectives. So I want to talk about how I started this study, because I'm not an infectious disease person. So I am environmental epidemiology person. So back to March 2020. Dr. Hu received an email from Dr. Mati Siren. Actually, he's a very old person. That's why his daughter sent the email. And Dr. Siren is one of the first researchers who identified this cadmium as a significant toxin in cigarette smoke. And so he reached out to Dr. Hu and asked about whether or not he's continuing research on this cadmium and lung disease based on our previous publication. So on the bottom, as you can see, so we have a publication about this urinary cadmium exposure and pulmonary function. And then we found that cadmium exposure is associated with the lower lung function. And also, Dr. Siren, read this paper. So this is very early study regarding COVID-19 and conducted in Wuhan. And then they, evaluate various risk factors for disease progression of COVID-19. And then they found that, so as now we know, so, older age and then other, the health condition are important risk factor for this disease progression. And one thing, very striking result, even though we know smoking is an important risk factor for, various disease and probably even including the infection, but this history of smoking, the odds ratio was 14. So one of the strongest effects they found in disease progression. So Dr. Siren asked Dr. Hu about, so the tobacco smoking is the most significant source of cadmium, even in the general population. So could there be a correlation between cadmium bioaccumulation in the body, either smoking or environmental exposure, and the way in which COVID-19 impact the individual patient? And another question is, could the amount of cadmium accumulation in the body be relational to the severity of symptom, to recovery rate, and conversely to mortality? So to address this question, so we need population with this data of exposure and outcome assessment. So one approach we can think about is using the data measured at the group level, so county level. So this is kind of the ecological data. And there are several studies published the association between this long-term air pollution exposure and COVID-19. And this study actually looked at this county level PM 2.5 concentration and then county level mortality rate. And then they found that this long-term air pollution exposure is associated with higher risk of COVID-19 mortality. So, but another one, especially for this, cadmium exposure, so we need data measured at the individual level. However, this exposure data, for example, these biomarkers of cadmium are very unlikely available in COVID-19 patients. So we thought this influenza virus infection and its severity of mortality probably, serve as a relevant proxy. So that's why we decide to look at this NHANES data, where these biomarkers of cadmium and mortality from influenza and pneumonia data are available. Before I show the results, so I want to give you some background of what is cadmium and why cadmium can, impact respiratory function and also, impact the infection. So cadmium is a toxic matter and very widely contaminated and then in our environment. So cadmium is it's naturally available, present in high amount in ore. And it is very widely used in industry, so anti-corrosion agent, so PVC stabilizer, color pigment, and rechargeable nickel cadmium batteries. So a few years ago, so as you can see in this picture, so this is the glass, provide to the keys at the McDonald's, and then this carton, this color. The cadmium were in that color, and that's why just drinking orange juice and water and cadmium can be exposed through this way. So that's why these glasses were recalled at that time. In general population, cigarette smoking is the major source, and actually for smoker, obviously cigarette smoking is the most important source of cadmium. For food is another source of cadmium. This is because the fertilizer, so the phosphate fertilizer, contain this, cadmium, and the plant can take up cadmium. So this is why the plant food have more cadmium than animal food, and the cereals and bread and tofu, soy product, rice, and leafy and root vegetable can contain this cadmium. And for non-smoker, food are the major root of this cadmium exposure. So then the next question is cadmium a respiratory toxicant? So the cadmium is very well known to increase reactive oxygen species production and the oxidative stress, and that's why they're leading to this local and systemic inflammation. And there are several studies showing that this cadmium, even at a non-cytotoxic dose, relatively low dose, can disrupt tight junction integrity in human airway tissue. And that's why impairing the epithelial barrier function. And another study showed this oral cadmium exposure, not the lung exposure, so inhalation, but oral cadmium exposure can promote this pro-inflammatory effect in lung tissue, and they're leading to higher susceptibility to bacterial infection. So then the next question is, can cadmium potentiate virus infection? So there are a few studies look at this cadmium and virus infection relationship. And one of them is by the Hu et al. Published in 2019. So they used the mouse model and then pre-exposure to cadmium through diet for 28 days. And they used this respiratory syncytial virus. And then they found that, as you can see here, so control group, cadmium group, and the virus infection, and then the cadmium and virus infection, and all these different measure inflammation measure. And as you can see, so in the group of having this cadmium and virus infection, so the inflammation level were higher and potentiate. So that's why this cadmium can potentiate a virus infection and then increase the inflammation in the lung, and then leading to lung injury. So this study suggests how the potential biological mechanism how this cadmium can work together with the virus and then leading to this lung inflammation. So the cadmium can dysregulate these metals and leading to oxidative stress, and then also disrupt some this metabolic pathway, and then leading to this T cell, the inflammation, and then cytokine the generation. And then finally, working together with this virus, and then lung inflammation and injury can happen. So another study, Chandler et al. So they also use this very low, relatively low chronic dose using the mouse model. And in this case, they use H1N1 infection. So this H1N1, as you may know, so this is the infection happened 1918 pandemic and also the 2009 pandemic, and then they found a very similar result. So for H1N1 infection, and then adding this cadmium can potentiate this inflammation effect. And finally, so that's why this cadmium exposure can enhance this inflammatory responses in the lung. From these two studies, I want to highlight, so they didn't use inhalation exposure route. They used this intake, so oral exposure, but this oral cadmium exposure, so which is similar to how general population, non-smoker can be exposed to cadmium, but that can also generate this inflammation and then impact the tight junction in the lung cell, and then leading to more worsening of the virus infection. So that's why, based on all this background, we ask about are biomarkers of this cadmium burden associated with the mortality related to influenza and pneumonia. And because, as I mentioned earlier, the smoking is a source of cadmium, so it's very difficult to disentangle cadmium from smoking. So that's why we want to make sure that this association between cadmium and mortality from influenza and pneumonia are independent of tobacco smoking, and then we use this NHANES data. Okay, so let's move to the actual study. So we use study data NHANES, so NHANES 3 and NHANES 1999 2006. So the reason why I cut 2006 is because there is no this pneumonia or influenza mortality case after the later cycles. So that's why I only include this NHANES 1999 and 2006. And also because the number of cases are very few below age 45. That's why I cut at age 45. NHANES 3, I use urinary cadmium because at that time, only urinary cadmium was measured. In continuous NHANES, I use blood cadmium. In this continuous NHANES, urinary cadmium is also available but urinary cadmium was measured only 1/3 of the population. And given that the number of cases are very small, few, that's why we only look at blood cadmium rather than urinary cadmium. And mortality data available through December 31, 2015. And we include, so we only look at this death from influenza or pneumonia, but not including other chronic lower respiratory disease. So we use this ICD-10, day 09 and day 18. So that's why we ended up having over 7,000 in NHANES3 and then 8,600 in NH99 and 2006. So statistical analysis, we use survey-weighted Cox proportional hazard model because NHANES is a complex sampled data. So we account for this complex sampled component. And we use attained age rather than time since baseline. This is because we are including a wide range of age, and at baseline, different age, especially this the influenza mortality, older group are much more susceptible to this infection and then die from this infection, and that's why their risks are different. So the time since baseline is not so recommended not to use that. This type of observational study, and then age is the more appropriate scale. So that's why we use this age as the time scale. To account for, because we are using urinary cadmium, so to account for urine dilution, we use creatine-corrected cadmium in NH3. So we developed two models. One is our main model. So we adjust for sex, race, ethnicity, phase, or cycle, and then education, BMI, hypertension, and serum cholesterol. And another model, because smoking could be important confounder, but it is also very difficult to disentangle cadmium from smoking. So that's why we want to be very careful. This smoking adjustment. That's why we made this as a separate model. But to also show that how the cadmium effect independent of smoking, we also did this stratified analysis and then look at only never smoker. But we are also so later I will be showing the result for the ever smoker as well. And effect estimate, we calculated hazard ratio and 95 confidence interval comparing the 80th percentile and then 20th percentile. So for urinary cadmium, so 80s and 20s are 1.2 and 0.34. So that's why this is equivalent to for 0.86 microgram per gram increase in urinary cadmium. And for blood cadmium, so this 80s and 20s is 0.8 and 0.3. So that's why this is equivalent to 0.5 microgram per liter increase in blood cadmium. So the results, so this is the characteristics of the population. So NH3, so over 7,000, that represents 66.6 million. Mean age was 60.7, so male and female, so well balanced. So over the median of 70.3 years, we identify 141 influenza and pneumonia deaths. And so that's why the incidence rate is 1.24 per 1,000 person years. And geometric mean of urinary cadmium was 0.48 microgram per liter. So in NH99 and 2000, so over 8,000, which represent 87.7 million US population. So the age and female, the mean age and female proportion are similar. And because this is a relatively shorter follow-up time, so we have only 56 death cases. And that's why incidence rate is 0.59. And this is that's why even though the base population sample size is larger in continuous NA, but because the number of the outcome cases are small, so that's why the NAs, this continuous NAs result could be underpowered. Geometric mean of blood cadmium was 0.46 microgram per liter. So this is the Kaplan-Meier curve, but this week, so environmental statistics week on Monday, Dr. Su's presentation with briefly talk about this during the Q&A session. So this is a Kaplan-Meier curve, but I used adjusted Kaplan-Meier curve and then create this adjusted cumulative hazard plot using the inverse probability weighting and divide this cadmium into low and high group, and then creating this plot if everyone is assigned to high cadmium or everyone is assigned to low cadmium, and what would be their probability, and then making this plot. So in NH3, as you can see, so the effect, so we see this exponential cumulative hazard plot, and then it began to differentiate around age 80, and the steeper the hazard plot can be seen in the high group over this low group, and the similar plot can be seen in 99 and 2000. But interesting thing is around age 100, so this low group, so red line is low cadmium group, so low group exceed the high group. But this is because at age 100, the very few people remain at risk, so that's why this is why this lower group actually exceed at a later age group, but age range. But overall, the high group have a relatively more steeper the cumulative hazard. And our main result, so hazard ratio. So this table is a little bit busy and complex, but the first two columns showing the NH3 result, and the next is NH99 and 2006. And then first, this side is showing the entire population, and then later stratified by smoker. So the middle part is never smoker, bottom part is ever smoker. And then two model, main model, and then smoking adjusted model. And then we found that in our main model, so hazard ratio in NH3, so urinary cadmium was significantly associated with this mortality from influenza, and hazard ratio was 1.15. But after control for smoking, this is actually expected. So the hazard ratio go down, and then we lose that statistical significance. In NH 99 and 2006, so the hazard ratio was 1.14. And interestingly, for blood cadmium, even after control for smoking, the hazard ratio remained almost identical. In never smoker, after we stratify, we found that even this hazard ratio is larger than in the entire population, so 1.27. And even after we control for this cotinine, which is the marker for environmental tobacco smoke, the hazard ratio didn't change. Instead, okay, so this means that for never smoker, so source of cadmium probably are independent. So even after we control for like a potential smoking effect, we see a cadmium effect from so other than smoking remain the same. And then continuous NHANES, so hazard ratio 1.7. Again, we still see relatively larger effect, and then those are the marginally significant. But for ever smoker, so we don't see an association. This is because probably the smoking, just smoking itself is a major effect. And then after we are controlling for this cotinine level, so this is another marker for the smoking, and then this hazard ratio go down, and especially for like a continuous NH from 1.14 to 1.03. So it's very difficult to disentangle actual. So if this is a smoking effect, and then this is an actual cadmium effect. And like a similar result after we divide this cadmium into tertile, and then I just want to highlight in tertile 3. So in NH3, so in the main model, we found that this tertile, so high tertile group, their hazard ratio was 2.29. So much larger than when we treat this cadmium as a continuous variable. And then after control for smoking variable, so this hazard ratio go down a lot, and then no longer statistically significant. And but in continuous NHANES, so not statistically significant, but relatively larger effects over. So hazard ratio was greater than 1. So because this is the environmental statistic week, so not only this epidemiologic finding, I want to briefly talk about some data issue and the statistical issue. And while I am working on this this project, I actually before I do this project, I didn't realize this issue in the use of public administrative data. But why I am doing this, and then during the the revision process. So one of the reviewer raised this issue, so data perturbation. I don't know how many people have heard about use of this public administrative data and the data perturbation. So there are two data perturbation incorporated into this NHANES. One is random noise was added to follow-up time and underlying cause of death. And second one is for older adults, age was top coded at age 90. So anyone whose age over 90, so 95, so we do not know what is their actual age. So all their age was coded as just 90 plus. And even continuous, and hence they lowered that top coding age from 85. So this is because for the like public data, especially for this much old age, so to protect some of this potential re-identification risk to the survey participant, that's why this administrative data they are putting this data perturbation. So then, how that perturbation can impact our causal inference and estimation of this hazard ratio? For this random noise to the outcome, so then that can generate some non-differential misclassification. That's why we can expect this bias toward the nerve. So that can be prevented if we are using restricted mortality data, but my study, I only use public data, so that's why this is unavoidable. But in the future, if I am using restricted data, then that can be prevented. For older age and the top coded age, because age-related mortality, even age 9 and older or 85 and older, we can expect within that age group, older people, so old in the older population, so older group, so even their risk could be higher. And for cadmium, we can expect, this is a cumulative matter, so that's why we can expect older people still contain more cadmium in the body. So that's why still within that group, so there could be confounding by age, and then they can bias this effect. So that's why we want to handle this issue. So to do that, so I just cut and then do sensitivity analysis after excluding this top coded age. And then I don't see any difference even after doing this. So which means that this top coded age and potential confounding by age effect is minimal. And then after we are accounting for this, and then we see very robust finding. Another statistical issue and epidemiologic issue is this competing risk. This is because in an event, so competing risk or competing event is an event in which the occurrence of one type of event preclude the occurrence of the other. So we can die from influenza, or we can die from cardiovascular disease, but we cannot die from both. So that's why these two outcomes compete each other, and that's why when we calculate cause-specific event, then this estimation can be invalid if we are not accounting for this issue. So that's why the ideal situation is estimating, what would have been hazard ratio in the presence of competing risk for other event, other causes. So we use this defined gray sub-distribution hazard model, and then CRR function in the R package CMP RSK. But the limitation is because this package does not support this survey component. So that's why we use this as just a sensitivity analysis, not as our primary analysis. So based on this analysis, hazard ratio in the presence of competing event, we found that in NHANES3, so this is a sub-distribution hazard model, and then cause-specific hazard model, we don't see much difference. A little bit, lower, but not much different between these two models. But in NHANES 1999 and 2006, you can see cause-specific hazard ratio was 1.23, but sub-distribution hazard model 1.15. So this suggests that our finding in NHANES 1999 and 2006 should be interpreted with caution, because there may be some competing risk. So, and then we did other sensitivity analysis, but the results were relatively robust. So we tried other urine dilution correction approach using more recent approach, but we don't see any difference. And additional adjustment for poverty income ratio, PEG years of smoking, and iron store marker, because the virus can use this iron. And also iron is important, the matter for the cadmium metabolism. So that's why we try to control for these iron store markers, but we don't see any difference even after controlling for this iron. And finally, we conducted some, if there are the effect of modification by age, sex, and race ethnicity, but there was no clear evidence. So in summary, so this is the first report that cadmium body burden, especially urinary cadmium in NHANES 3, may be associated with higher risk of mortality from influenza or pneumonia among older adults. And that this association remains statistically significant even among never smokers. The cadmium may predispose individual to worse pulmonary disease from this respiratory infection, no matter where this cadmium source come from, the cigarette smoking or contaminated food intake. And as I mentioned earlier, so potential biological mechanism, so cadmium can accumulate in the human lung tissue and also interfere with this antioxidant defense system. And that's why potentiate the cytokine production, lung inflammation, impair macrophage, immune function, and then disrupt this tight junction integrity in human airway epithelial tissues. So the next question, because what I am showing is not COVID-19, the question is, can cadmium worsen outcomes from COVID-19? Probably, given this shared biological mechanism between influenza and COVID-19. And also cadmium is an important toxicant of tobacco smoking. And as I showed earlier, so smoking and COVID-19 disease progression, ARDS ratio 14. So smoking is known risk factor for COVID-19 severity and mortality. And also, biologically, so a few studies show that the smoking upregulate angiotensin converting enzyme 2, ACE2 receptor. So this is still some controversial, but this is known as a adhesion site for COVID-19. But actually, there is no data available, the association between this COVID-19, the cadmium and ACE2 receptor. So this is why epidemiologic and experimental studies are needed. So this study provides new evidence that this population can benefit from this lowering cadmium burden when the next pandemic occurs. But as we know, that it's very difficult and that cannot be done suddenly, and then takes time through this policy change to lowering this cadmium in the population level. So tobacco control, ambient air quality, food contamination reduction. Good news is since this 1988, so there is some feedback. Can you please mute? Yeah. Okay. So this population cadmium concentration level has been reduced. So this black line is NHANES 3, and then the later cycles of NHANES. So this population urinary cadmium level shifted toward the lower level, which is good. So which means that mainly this is through the tobacco control, and then the population cadmium level become lower. But even continuous NHANES cycle, there are tons of studies showing that current population level of cadmium are associated with many different chronic diseases. So then primary prevention strategy at individual level, obviously stop smoking, and even e-cigarette. And the e-cigarette and then not only cadmium, but also other matter has been reported. And then, as I showed earlier, so food are major source of this cadmium. And then, very recent study using this NHANES data, they show, different types of food can be major this cadmium contribution. And as you can see, so different race ethnic groups, so different types of food are major source. So the lettuce, so white and black, and the Hispanic tortilla, and then rice for others, and mainly including Asian group. So it is important to acknowledge what are important major source of cadmium in food. And I want to be very careful to mention the secondary prevention, because this day there is very anecdotal information regarding how can we boost our immune function and then prevent this COVID-19. So this is why this is just one of the few scientific findings showing and how antioxidant can mitigate cadmium toxicity, but this is not direct evidence for COVID-19. So please be careful. But N-acetylcysteine, which is very well-known antioxidant, in this experiment, they show that the NAC can mitigate cadmium toxicity even in lung tissue. Finally, I want to talk about, this is the known, this COVID-19 severity risk factors, the comorbidity, chronic disease, and then some lifestyle and environment. Those are important potential risk factor. And now we know that this, ambient pollution, industrial wastewater, smoking, food are the major source. And then cadmium can also, a lot of studies have show that this cadmium associated with these various chronic disease, and then finally that can also impact the susceptibility to the COVID-19. So that's why this pandemic actually add this cadmium, and not only cadmium but also environmental factor. Why we need to control and then reduce population level to prevent not only the chronic disease, but also this chronic condition can also impact the severity and the mortality to COVID-19. So this is why environmental the health is important to promote general population health and public health. But cadmium is not the only one. So many more environmental toxicants can also impact inflammation and the immune function, and eventually increase the susceptibility to COVID-19. So this is why it is important to identify those modifying factor, risk factor, especially given that this continued absence of antiviral agent with this uncertain timeline of vaccine development. Okay, so I'm done. So thank you for your attention, and I will be happy to answer. And actually, I want to open not only just I am talking, but also from the audience, and hopefully we can have some active discussion. So, thank you very much.
Thank you, Sanyang. That is a very good presentation. And yeah, those have very great questions for our environmental health scientist. So any questions from our audience, please unmute yourself, and we welcome all the discussions at this point. Thank you.
Hi, Dr. Park. First, that was a great presentation. Thank you for that.
Hi, John.
I think it raised a lot of really important, questions kind of for future research as well, kind of regarding the social and environmental contributors to, severe respiratory infections. So I don't really work in environmental epidemiology very much. I was hoping you could kind of comment a little bit more about kind of how cadmium accumulates in the body. I saw there were a couple points that you had in your presentation about that, where it accumulates in the lungs, but I'm wondering how quickly does it accumulate, and kind of how long does it last and how long does it, seem to kind of contribute to, having these adverse health effects?
So the cadmium. So as I showed earlier, so cadmium can be exposed through either like a cigarette smoking or ambient air. So that's why through the inhalation, or through the intake of this contaminated food. So that's why it can go to the lung or in the gut, and then because our body have some the transporter. And that's why those cadmium can move around and then move to the circulation system, and then it ended the renal, so kidney, and then can be excreted. But the cadmium is one of the matter chemical that it's very difficult to excrete and get out of the body. So that's why cadmium stay in the kidney very, very long time and reaccumulated and then reabsorbed and then move around again. So this is why our body have not evolved to to handle this cadmium and then get out of the body. So that's why cadmium occurs in the body. So for like a urinary cadmium, it is known that urinary cadmium can reflect several decades of exposure. So this is why we are using urinary cadmium as a potential biomarker for like a chronic cumulative exposure. But for general population, even blood cadmium can also reflect relatively long-term exposure. So this is why cadmium can stay in the body very long time, and then especially for older people because they have been exposed for a long time, and then so that can impact various chronic disease, including cardiovascular disease, respiratory disease, and diabetes, and in cancer as well. So cadmium is one of the known carcinogen, and we haven't actually focused on this infection and the virus infection and how this environmental chemical can impact it. And that's why this is even though the COVID-19 is a very serious public health crisis, but it is another like a good opportunity for us to think about how environmental exposure and then chemical that have been we only consider for just chronic disease, but it can be also important for like an infectious disease. And COVID-19 is not just a single event, so we expect this type of pandemic happen in the future again.
Thanks.
Dana, do you have a question or comments?
Hey, I see there's also a few questions in the chat, so that's great. So actually, this is a good follow-up from John's question, because I was just thinking about, age, and, you answered this really well about cadmium accumulating over time. And so that may be one of the reasons why you're seeing higher susceptibility in older aged people. But in your analysis with NHANES, and it might've been hard to do with NHANES, did you see associations with younger people in cadmium as well?
So as I showed you before, this effect modification. So and this is because even this NHANES 3, so we have median follow-up time is 17 years. So even 17 years, the number of cases is only 141 cases. That's why it's very difficult to evaluate this disease if after stratify by different subgroup. And that's why I didn't look at age under 45, but 45 and 64 and then 65 and 89 and then as you can see 90. So because there are very few people in that group, that's why the confidence interval is very wide. But we see that after age 45, which means that so they have been exposed to cadmium for relatively long time. It's already a long time. Even within this age group, they are younger. But then, given this accumulation of the cadmium, then it can also impact, if they get infected, then this cadmium probably do something. And so we don't see much difference between this 45 and 64 and the 65 and 89. And we see that 90 and older, so hazard ratio is much higher, but because the confidence interval is very wide. And interesting thing is when we look at blood cadmium in continuous NHANES, we don't see an association at this age of 64, 65 and younger. And then this is null. And only 65 and 84, and then 85 and older, we see their hazard ratio is around this similar level. So, but this is probably the biomarker difference, and then also this continuous NHANES is the number of cases is too small, so it's not easy to like generalize if younger people really less susceptible to compared to the older people. But if we weigh more in urinary cadmium and longer follow-up and then larger number of cases, then I don't see difference between these different age group.
Thanks for showing that again. And it's also very interesting how a change in a biomarker from urine, blood to urine can really change the way you can evaluate this question using the publicly available data. Thank you.
Sang-Yang, I think there are a couple of questions or comments in the chat. Do you want to?
Actually, I don't see the chat, so that's why.
Okay. So Trish has a question.
Trish, here, okay, let me, so I just opened the chat box.
Okay.
Question is secondhand smoke available in NHANES? The control for secondhand. Yeah, so the secondhand smoke, so even though the serum coating level, so serum coating level can capture this environmental tobacco smoke, not only for active smoker, but also for passive smoker and any like even third hand smoke, and then get into the body, so that can capture. So I try to even control for those the serum coating level, and for the never smoker group. So this coating adjustment didn't change this cadmium effect, which means that if cadmium source is not from main source is not smoking, then cadmium have a independent effect, and even after control for potentially the secondhand smoke. But for the current smoker and then ever smoker, the coating is very difficult to, this is an active smoke or this is a secondhand smoke. So in that case, after control for this coating, we lose all the association. And then, so Marie also mentioned this coating was the, and then what for future study of COVID, what might be the implication for secondhand smoke cadmium during stay-at-home period that may differ from previous years? This is interesting because I don't know. So if the stay-at-home, so stay-at-home order and also the our lifestyle have been changed a lot. So not only this. The exposure to different types of chemical. So when we are outside and then when we at home, so depending on the amount of time you are staying in one place, and then probably the major contaminant in your home. Whereas in the past you probably are exposed to this type of chemical from many different sources, and also. I think that this COVID-19 and the mental health is important, like a consideration we want to consider in the future. Not only just the source, but also the, just staying at home and then our mental health. We are very susceptible to this one, and then how this stress modulate this, this chemical toxicity and then that can impact the susceptibility to this the virus infection and others. So that can be another area we wanna evaluate in the future. So not only the question, but also is 453. But from the floor, anyone who have any insight about this, like a cadmium and COVID-19, but also other chemical exposure, and then how, what can we do in this the pandemic era, and this may not be just one time pandemic. So COVID-19 and then other similar types of this virus infection could happen in the future. So then what can we do? So I also want to hear your insight about so over the past several months, so what you have thought about this issue. Anyone? Or anyone who are doing directly, because this is an environmental statistics week, but we invite speakers who are very actively doing this COVID-19 research, and so that's over the past 3 days. The leading scientists who are doing this COVID-19, they are showing fantastic results. But in this environmental health community, still I don't see what kind of research and how can we respond to this COVID-19 in terms of this environmental exposure. So if anyone who are currently doing directly related to this COVID-19 and some of the environment and infection interaction, then I think this is a good time that we can share some this idea and insight that can also advance our understanding.
Yeah.
Kyun?
Yep.
Yeah, can you hear me? This is Marie.
Marie,
I just want, so I just wanted to say, I think that's a really important question. I think one of the things that, we've noticed is the health disparities with the impact of COVID-19. And I think, one way that environmental health sciences can enter in that discussion is that we already know there are, great differentials in exposure by race, ethnicity. And I think, the contribution of that information could really help to minimize some of the burden. That's one potential angle that we can take.
Yeah, I agree. So health disparity issue is another domain that we need to think about this. And, this environmental disparity and the differential disease development and the disease incidence by this exposure. And so that's why this is not totally separate issue. So this environmental exposure and then differential. We used to only look at just chronic disease, but this is all are connected, and then that lead to push those environmentally disadvantaged population, and then they could have higher disease, and then that can also make them more vulnerable to this infection. And so this is why I think this is also important consideration we need to think about.
Dr. Park, this is John again. So I just wanted to, add my kind of 2 cents from the infectious disease side of things. For somebody like me that has kind of very limited experience with environmental epidemiology, this is really, an interesting kind of perspective to take. And kind of your comments or just kind of in response to your question about how to, kind of address the current COVID pandemic, I just want to say, I think it's important not to also lose sight of just the yearly, non-pandemic toll from, respiratory viruses like influenza or RSV is still really substantial. And so kind of looking at these environmental mechanisms, I think, can have really long-standing benefits. And I think the questions that you raised in this analysis are really, really exciting and interesting. So.
Thank you, John. And actually, congratulations your defense. And I missed your defense because I had another meeting at the same time.
Oh, thank you. Yeah, no problem. I appreciate it.
Okay, so the Lu need to leave, and I think so. This is time, so we have to stop here. And so, thank you for coming to this talk. And so, we have one last this environmental statistic talk tomorrow. So please join again, and please stay healthy and see you around. Bye.